So active transport, as opposed to our passive transporters that just allow things to go down their concentration gradient. Active transport, on the other hand, we are sending things up the concentration gradient. And to do so, we have to use energy. All right. So same principles as passive transport in that you have to have a membrane protein. It has to be able to move things from one side to the other. But for active transport, we're moving against a concentration gradient, going up from a lower concentration to a higher concentration rather than going from a higher to a lower. And that's why we need energy, because the process has a positive delta G associated with it, which means it's non-spontaneous. To make it spontaneous, we got to add energy. This is why a lot of these transporters are called ATPases, because they use ATP directly for energy. When they do that, we call that primary active transport. Utilizing ATP to drive something, a particle, usually for ATPases, it's going to be an ion, against a concentration gradient. And the first example we'll talk about is P-type ATPases. These are cation transporters, reversibly phosphorylated by ATP as part of the transport cycle. And this causes the P-type ATPase to basically move between two conformations, one where it's phosphorylated and one where it's dephosphorylated. The phosphorylation of the P-type ATPase causes a conformational change, which triggers the movement of the cation across the membrane, whichever way we're pumping it. And the directions and the number of ions vary depending on which P-type ATPase we're looking at. We're going to look at two types of P-type ATPases. But these ATPases, these transporters, use primary active transport. And again, primary means that the transporter is using energy directly, typically in the form of ATP, though not always. But for our examples, it will be ATP that it uses. So there's lots of different types of P-type ATPases. They can vary depending on where they're located, where they're expressed in the body, what type of ions they pump. But there's at least 70 types encoded by the human genome that we are aware of. They all have homology, meaning they come from a progenitor ATPase that became all of these different ATPases. So they have similar folding, similar shapes, same domains, but they vary depending on which ions they recognize and how they pump them or where they're located. For our P-type ATPases, asparagine is the residue that's phosphorylated, and that happens in the phosphorylation domain, or the P domain. And I would like you to know that there are 10 alpha helices spanning the membrane for our P-type ATPases. So this shows the different domains in the P-type ATPase. The biggest one is the transmembrane domain. That's the one that gets us from one side of the membrane to the other. So this is the part of the protein that the ions are going to pass through. Down here, the green, kind of the toe of the boot, this kind of looks like a boot. If this is the ankle, the heel over here, the green part would be kind of like the toe. And this is where we bind to ATP. Note that we're facing the cytosol. The ATP, or nucleotide binding domain, is located on the cytosolic side. The reason why is because ATP is going to be inside of our cells, not outside of our cells. So the energy needs to be provided from the cytosol. ATP binds to the nucleotide binding domain. It then passes a phosphate over to the phosphorylation domain, or the P domain. That causes a conformational change in the protein. This conformational change is conducted through the actuator domain to the membrane domain. We need that because we just have to connect these two domains, the P and the N domain, to the M domain. No, I said a lot of letters there. So let's go over it again. So when the transporter is ready to go, in this case, we're looking at a calcium P-type ATPase, which we're going to look at an example of here in a sec. You can see the calcium ions need to be in place, ready to go, ready to be transported from the cytosolic side to the lumen side. Then ATP binds, phosphorylates the asparagine in the P domain, which causes a conformational change, which is conducted through the actuator to the membrane protein, and this triggers the conformational change that will move the calcium over to the lumen side and then release those calcium ions in the lumen. We need energy because there's more calcium in the lumen than there is in the cytosol. Because there's more calcium in the lumen, it costs energy for us to move more calcium across, and that's why we have to bind to ATP. So you need to know what each of the domains are, their names, and what they do, and be able to recognize them if I showed you a picture of one of these proteins. You need to be able to identify where the domains are located. It would be this same picture here. I wouldn't use a different picture. Okay, our first example of a P-type ATPase is going to be the sodium-potassium pump, maybe the most well-known P-type ATPase. You learn about it when you learn about action potentials in biology, for instance. And all animal cells maintain a gradient of sodium and potassium across that outer membrane, our plasma membrane or cell membrane. And this gradient is generated by the sodium-potassium pump, and it does this by pumping sodium out from the cell and potassium into the cell. So the sodium is going from the cytosol out to the lumen, potassium coming from the lumen into the cytosol. It's really critical that we form these gradients because we need them to allow for the cell to be able to do what it needs to do. So it's really important. We'll see later on when we look at the sodium symporters, they utilize the gradient generated by the P-type ATPases. If you go and learn about action potentials, which we're not going to cover in here, but action potentials also rely upon this same gradient. Let's go ahead. All right, so this is a flipped picture of a P-type ATPase without all the detail, obviously. We have the transmembrane domain here, then we have the portion that binds to the ATP, so all those other domains are going to be represented in the big part here. Notice that the portion with the ATP binding domain is facing the cytosol, which it needs to do. The other side is facing the lumen. And we can see for every ATP that we convert to ADP plus an organic phosphate, we pump 3 sodium out from the cytosol to the lumen, and we pump 2 potassium in. When we do this, we generate obviously a chemical gradient for both sodium and potassium. Again, sodium's concentration will be higher on the outside, lower on the inside, vice versa for potassium. But we're also going to generate an electrical potential with more plus charge on the outside, more negative charge on the inside. Both of those things are generated from these sodium-potassium pumps, and both are really important for the overall functionality of the cell. Okay, so the calcium pump, as I mentioned, pumps 2 calcium per phosphorylation. The sodium-potassium pump, we're going to see that here in a second once again. The sodium-potassium pump moves 3 sodiums out and 2 potassiums in per phosphorylation. Both ions get pumped against their concentration gradients. And when we are in the E1 conformation, that means that the binding sites for the ions are facing the cytosol, and E1 also has a higher affinity for sodium, which is going to bind when it's facing the cytosolic side. When you phosphorylate the transporter, then it's going to face the outside, that's the E2 conformation, which is going to have a lower affinity for sodium, but a higher affinity for potassium because it has to bind the potassium on the outside. So you can see that process here. So again, here are the domains. I'd expect you to be able to recognize the domains on this picture as well, by the way. And you can see this is the N domain, nucleotide binding. This is the P domain, the phosphorylation domain. And over here is the actuator, and then the membrane, transmembrane domain at the top. So we can see we have binding sites for 3 sodiums on the left. There are circles, and the squares represent the potassium. When we're in the E1 phase, or the E1 conformation, we're facing the cytosol, and the binding sites for sodium are going to have a high affinity for sodium. The ones for potassium are low because it just spit out the potassium into the cytosol. So what happens is the sodiums get in place and they bind, and the ATP comes in and it binds as well. And then we get the phosphorylation of the P domain and generation of ADP, which then is removed from the binding site. When this happens, we get the conformational change from E1 over to E2. When it's in the E2 conformation, it no longer wants to bind to the sodium, so it spits the sodium out, and it's going to be over and released on the lumen side of the transporter. And now it has a high affinity for potassium, so potassium comes in from the outside and binds to the binding sites on the E2 conformation. Then we dephosphorylate, and so the phosphate is removed through enzyme activity, and that causes us to shift back to E1. We're now facing the cytosol again, and those potassiums are released. Once that happens, we reset back to where we started. So know the way that the conformation changes, what happens to the affinity with sodium and with potassium as we go through the conformational changes. Know that the steps when the conformational change happens, which is when we phosphorylate. When we get the conformational change, we change from being open to the cytosol to being open to the exterior and the lumen. And that's when we release our sodium and bind to our potassium. And then we go back to E1, potassium is released. Now we can bind to sodium again. So definitely know the progression through this overall process. Calcium pump works pretty much the same way. It's going to have the same structure, same regions, domains, all that good stuff. But it's going to bind calcium on the cytosolic side, and it's going to pump it to the lumen side. Calcium pumps are found in the sarcoplasmic reticulum of muscle cells. That's the ER, what we call the ER when we're in muscle cells. It turns out that we store a lot of calcium in there, in the sarcoplasmic reticulum, by pumping it out of the cytosol into the SR. We do the same thing, actually, in our endoplasmic reticulum in non-muscle cells, but we have more calcium typically in our sarcoplasmic reticulum. And this is tied to the physiological process of muscle contraction. So when you get the signal to contract muscles, that triggers calcium release from the sarcoplasmic reticulum, and that triggers the muscle contraction. But then we got to pump the calcium back in once the contraction's over. We got to get the concentration of calcium back down to that lower level. And it stays very low in the cytosol, 0.1 to 1 micromolar concentration, whereas in the sarcoplasmic reticulum, it can be high as 100 millimolar. That's a huge difference there. That's about 5 orders of magnitude, or almost 6 orders of magnitude. So big difference in the concentration from the cytosol, where it's really low, to the sarcoplasmic reticulum, where it's really high. We also have calcium pumps in the plasma membrane that pump from the cytosol out to the lumen there. And same thing, just trying to keep that cytosolic concentration lower so that the signals work when we pump our, or when we contract our muscle. So it's going to have the same mechanism as we saw with sodium, the sodium-potassium pump. It's going to switch between E1 and E2. In the case of the calcium pump, the E1 state, it's going to be facing the cytosol, and that's when calcium is going to bind. So it's going to have a high affinity for calcium in the E1 state. In the E2 state, that's when the binding sites will face the sarcoplasmic reticulum, and we want to release the calcium at that point. So in E2, it's going to have a lower affinity for calcium. Same process otherwise. ATP binds, we phosphorylate the calcium pump. It's going to cause that shift from E1 to E2. The calcium gets pumped out, and then we dephosphorylate and switch it back to E1 again. And again, we have to release the ADP, release the phosphate once we reset back to E1, and then we're able to go through the whole cycle again. So it's the same thing we saw for E2 and E1 in the sodium potassium pump. We have our calcium sites. When we're in E1 state, we're open to the cytosol. Our calciums come in and bind to the binding sites here. Then ATP comes in and binds to the nucleotide binding site. We phosphorylate the P domain. That triggers the conformational change from E1 to E2. Once we do that, now we're facing the lumen rather than the cytosol. Calcium's released due to a drop in the affinity for calcium when we're in the E2 state. That allows them to be released. The phosphate gets removed by an enzyme, and we switch back to the E1 state, and we're ready to do it all over again. Once again, the pumping is happening from inside in the cytosol out into the lumen if it's in the plasma membrane. If it's located in the sarcoplasmic reticulum, once again, the cytosol will be outside. The inside will be the lumen of the sarcoplasmic reticulum. So lumen here could be either outside of the cell or in the sarcoplasmic reticulum. So this is just a comparison of the tertiary structure of the sodium potassium pump, which is represented in gold. The calcium pump is represented in blue. And what you can see is they are very similar. Same alpha helices, same positions, but some differences. You can see that they're not exactly the same. There's some subtle differences here and there, and you can see that they don't line up perfectly. But definitely you can see the relationship between them and that they look really similar. And that tells us, of course, that they come from a common precursor. But we can also see that from looking at the sequence of the protein itself and doing homology. And we can see that there's a lot of homology between these receptors as well. Okay, now let's take a little break and talk about our F-type ATPases.